Hi, I'm Kirsten Andreatta from Gilead Sciences. Thank you for this opportunity to present results from our long-term Bictegravir and Dolutegravir resistance selections initiated with HIV-1 containing M184V in reverse transcriptase. M184V is a common NRTI resistant substitution, conferring high-level resistance to lamivudine and emtricitabine and increased sensitivity to tenofovir and AZT. Since 3TC or FTC is a component of most ARV regimens, M184V occurs in up to 67% of people after treatment failure and has been detected in 10% of virologically suppressed clinical trial participants. M184V can also be transmitted and is detected in about 4% of acute HIV infections, but only in 1% of chronic untreated infections due to reversion to wild type in the absence of selective pressure. The INSTIs Bictegravir or BIC and Dolutegravir or DTG have high barriers to resistance, and previous studies have suggested that the presence of M184V prevents development of DTG resistance. For our study, we sought to assess BIC and DTG resistance pathways in HIV with pre-existing M184V by performing parallel dose escalation selections in MT2 cells using BIC and DTG as well as raltegravir or RAL and elvitegravir or EVG as controls. First, in the RAL and EVG controls, the selections progressed at similar rates. In these experiments, drugs began at 1x the EC50, and when viral breakthrough occurred, the supernatants were harvested and used to infect fresh cells at twofold higher drug concentrations, which we refer to as increasing the passage number, abbreviated here as P. Starting with the M184V mutant, RAL selected Q148R plus other substitutions, and EVG selected T66I, both of which are the same resistance patterns that emerge in wild-type infections. The mutations associated with INSTI resistance are shown here in bold. The final viral pools were tested at Monogram Biosciences, and the high-frequency substitutions were confirmed by population sequencing. The expected resistance profiles and reduced replication capacity based on the integrase gene were also observed. Moving on to BIC and DTG, we started 2 independent selection experiments for each drugs, the second of which we split into 2 cultures halfway through, which we call 2A and 2B. Overall, BIC and DTG took longer to step up drug concentrations than RAL and EVG, consistent with BIC and DTG having higher resistance barriers. And the BIC and DTG selections were continued for over 200 days. Remembering that M184V was a full mutant in the starting virus, and there was no selective pressure for maintaining this substitution, we also wanted to see if reversion occurred during the course of the experiment. What we found was that M184V persisted as a full mutant throughout the entire selection for all drugs except RAL, which was the only selection where we saw reversion to wild type. Looking at just the first set of BIC and DTG selections, both progressed at similar rates. By deep sequencing, at passage 4, we saw no changes in the BIC selection and emergence of S153Y and M154I in the DTG selection. S153Y is a known substitution associated with low-level resistance to INSTIs, and M154I is a polymorphic substitution not associated with resistance. By passage 6, S153Y had also emerged in the BIC selection, along with V72I, another polymorphic substitution. And by passage 10, most of these changes had become the major variant in the culture. By the final passage, BIC had also acquired G140E, and DTG had G149R. In these viral pools with S153Y, we see low-level decreases in BIC and DTG susceptibility, with fold changes 1.6 to 1.8-fold over wild type, but both drugs are still considered active. We also saw low-level resistance to EVG that was above the assay cutoff and reduced replication capacity. In the second set of BIC and DTG selections, as I mentioned, we split each into 2 sister cultures after we saw the first genotypic changes. By passage 7, the BIC selection had acquired R263K, which is the signature resistance mutation selected by both BIC and DTG. By passage 6, the DTG selection had acquired an N144D substitution at low level, which I've highlighted here in purple because it's not previously been associated with INSTI resistance. We'll look further at each selection on the next slides. Looking at just the second set of BIC selections, at passage 8, both sister cultures had R263K, and one had additional substitutions M154I and R262K, which were observed at passage 9, the final passage. As I've mentioned, M154I is a polymorphic substitution, but R262K is a novel mutation not previously associated with INSTI resistance. In the second set of DTG selections, the novel N144D mutation emerged at passage 6 at low frequency and became the major variant by passage 8. By passage 10, both cultures acquired M154I. One of the cultures also developed the R263K resistance substitution and A265T, which is a polymorphic site. In the other culture, the secondary INSTI resistant substitution E157K was acquired. Now going back to the second set of selections side by side, the integrase genotypes by deep sequencing are summarized in the top right. In the viral pools from the BIC selections with R263K, we again saw minor decreases in both BIC and DTG susceptibility, below the assay cutoff for a resistance assessment, and resistance to EVG. The DTG viral pools that contained N144D also showed low fold changes in BIC and DTG susceptibility and resistance to EVG, further implicating this substitution in drug resistance. Next, we built site-directed mutants to further characterize the substitutions that emerged. In the first set of selections, which both went down the S153Y pathway, we can see that mutants with S153Y had low fold decreases in susceptibility below the assay cutoff for BIC and DTG, and the other substitutions did not seem to have an effect, except for the triple mutant of S153Y plus V72I plus G140E. G140 is a site where we see secondary or accessory INSTI resistance, but this is usually an A, C, or S substitution. In the selection, G140E was only detected in 10% of the sequences in the final passage, and the replication capacity of this triple mutant is less than 1% compared to wild type, which suggests this virus may not be viable in vivo. Also, because of this low replication capacity, we'd like to confirm this phenotype with additional experiments. We also built site-directed mutants representing the substitutions observed in the second set of BIC and DTG selections. First, looking at the R263K mutants selected by BIC, we saw small phenotypic shifts to BIC and DTG that increased to intermediate resistance when R262K was added. As for the novel N144D substitution selected by DTG, we also saw small phenotypic shifts to BIC and DTG and some resistance for EVG and RAL. When N144D was combined with other INSTI resistant substitutions, R263K or E157K, or both the M154I and A265T polymorphisms, we saw higher resistance to all INSTIs. But again, these combinations were present at low frequencies in the selection, and the replication capacities are extremely low, so we'd like to confirm using another phenotypic assay. Finally, we also sequenced the NEF gene in all of the BIC and DTG selections, because Malay et al. In 2017 described changes in NEF, notably the 3' Polypurine Tract, that resulted in high-level resistance to all INSTIs. We did see 2 changes in the 6G pattern of the PPT in 3 selections at low level. We built these mutants, including the original one described by Malay, and preliminary results found no resistance to INSTIs or any other antiretroviral drugs tested. But this requires further confirmation. In conclusion, HIV with M184V and RT acquired INSTI resistance substitutions under long-term selective pressure with BIC or DTG, going down the known resistance pathways of S153Y or R263K and a novel resistance pathway N144D. The viral pools and high-frequency variants retained sensitivity to BIC and DTG as assessed by the phenotypic assay cutoffs, but complex integrase substitution patterns, only present as minority variants, demonstrated broad INSTI resistance. However, low replication capacity suggests possible non-viability in vivo and confirmatory phenotyping is planned. M184V was maintained throughout all BIC and DTG selections, indicating that M184V does not protect against INSTI resistance development. And finally, our data support the need for continued surveillance of novel INSTI resistance mutations, both in vitro and in vivo. Thanks for listening.